Welcome back, friends. So in this video tutorial we will going to talk about the classical pathway. So this will going to be the third video lecture of complement system. So I have already uploaded 2 more videos on that. So if you are the first in this channel, then I would like to recommend you please go and watch the previous 2 video, because those 2 video will help you to understand about the concept of the classical pathway. Okay, so let's start. So we have named classical pathway because it was the first complement pathway to be worked out here, but that is not important thing. The important thing is classical complement pathway starts when a antigen bind with the antibody, okay, and leads to the formation of antigen antibody complex. Clear? Now the thing is, I have already told you in the first video of complement pathway, that is antibody have 2 part. One is FAB part, another one is FC part, and the antigen will bind with the FAB part of the antibody, just like that. Okay, so when this kind of complex is formed, the component of the classical pathway can bind with that FC part, okay, and start the entire classical pathway. Okay, so the first important criteria for the classical pathway to begin is the formation of antigen antibody complex, and that is why we have told that classical pathway is the effector pathway for the humoral adaptive immune response. Okay, I have already discussed that immune system can be divided into 2 parts. One is innate and adaptive can be further divided into 2 parts. One is humoral adaptive immune response, another one is cell mediated adaptive immune response. Cell mediated adaptive immune response mainly involves the function of T cells, and humoral adaptive immune response mainly involves the function of antibodies. Okay, so remember, whenever antibodies are involved, it is an example of humoral adaptive immune response. And in many cases you can also see that humoral adaptive immune response and cell mediated adaptive immune response works together. We will also going to discuss about those things. Okay, so that is the idea. So formation of antigen antibody complex is important for the starting of classical pathway. Okay, along with that, so now it is important to talk about what are the function of antibodies. So antibody can destroy antigen in various ways. One is neutralization, that is here antibodies main focus is to inactivate the antigen, right? Second one is the opsonization. Here the main focus of antibody is to facilitate the phagocytosis of antigen. Okay, along with that, another important thing is to activate the complement. That is our topic of discussion, right? Because if the complement pathway is activated, it will facilitate the lysis of the cell. How we will going to talk about. Okay, so along with the antibody, there are others activators also present for classical pathway. Okay, they are viruses, necrotic cells, subcellular membranes of mitochondria, aggregated immunoglobulin, beta amyloid found in the Alzheimer's disease. Okay, so you have to remember all these names, because MCQ may came from that things, that they will told you that how the classical pathway can be activated, and they will not give you antibody as an option. Instead of, they can also give you those things as option, then you have to find out. Okay, so now I have told you that antibody can activate the complement when its FAB portion is in interaction with antigen. Okay, now here I want to mention that here I want to mention that antibody can be divided into 5 parts. Okay, and there is a mnemonic I would like to recommend you, please follow this mnemonic, because it will help us to understand the antibody chapter, because there are various type of things that we have to remember, and we will going to easily remember it with the help of this mnemonic, that is game D. Okay, game, I think you can remember the game along with D. Okay, so G for IgG, A for IgA, M for IgM, E for IgE, and D for IgD. Okay, this is the 5 type of antibodies. Among them, Ig can be further divided into Ig1, Ig2, Ig3, and IgA, that is our secretory antibody, can be further divided into 2 parts, that is IgA1 and IgA2. Okay, we will going to talk about those things in details later. Now, among these 5, only IgG and its subtypes and IgM can activate complement. Why is this so? Before that, I want to talk about the order of the ability of the antibody to bind and activate complement. So IgM is the most efficient antibody in that case, then Ig3, then Ig1, and the power of Ig2, IgG2 in that case is least. Okay, so IgM, Ig3, Ig1 much less than Ig2. Okay, now the first component of the classical pathway is C1. Okay, so I have told you in the previous video that first one component of the pathway will be activated, it will activate the next component, then the next component will activate the second next component, and that is how the entire thing is going on. Okay, so here the first component is C1. Okay, the C1 will going to bind with the Fc portion of the antibody. Okay, now C1 is a complex that consists of 3 different proteins. One is C1q, C1r, and C1s. The C1q consists of 18 polypeptides arranged in 6 identical subunits. Okay, we will going to see the picture of C1 also. I think you can understood it very well with the help of picture. So C1 consists of 6 identical subunit. These 6 identical subunit have 18 different polypeptides that I have not mentioned here. So this is the part of the C1q. Okay, and in between them there are various type of other protein present, that is the C1r, this one is the C1s, it is again C1r, C1s, C1s. So what are they? They are serine protease. Okay, because in the previous video I have told you that protease will going to activate the complement components. Okay, so they are the protease here. Okay, so another important thing, as you can see, one molecule of C1 can bind with 6 Fc portion of the different antibodies, right? That is the important thing you have to remember. Okay, and another important thing, C1 cannot work in absence of Ca2 plus. It requires Ca2 plus for its activation. Okay, otherwise it cannot perform its work. Okay, so we have learned that C1q will first react with the Fc portion of the antibody. Okay, and C1r and C1s here will play the role of the protease here. Okay, now here lies the answer why only Ig and IgM can activate complement, IgA, IgE, IgD not, because IgA, IgE, IgD lack C1q receptor, and that is why it cannot bind and activate C1, and that is why it cannot activate classical complement pathway. Clear? So now let's move to the steps. So as I have told you, first step will be the formation of antigen antibody complex. Okay, so in that surface there are various epitopes present. Now the question is, what is epitope? Epitope is the specific part of the antibody, specific part of the antigen that can interact with the antibody. Okay, so these parts are epitope. Okay, okay, so that is how antigen antibody complex is formed. Now once antigen antibody complex is formed, this antigen antibody complex, the Fc portion of that antigen antibody complex will interact with the heads of the C1q. Okay, as you can see, they are interacting. Okay, and this interaction will activate C1R and C1S, and I have told you C1R and C1S are serine protease. Okay, now the C1R upon activated, C1R after activated, it will cleave C4. This is the second component of your classical complement pathway. Okay, and breaks them into C4b, C4a. Now please tell me that what part will be larger and which one will be smaller. I hope you can guess. So C4b is the larger part and C4a is the smaller part. So C4b will take part in the further reaction and C4a will dissociate. Right? Now this C4b will bind with the C2, okay, and expose C2 to the action of C1S, that is the second serine protease. Okay, and this serine protease will break C2 into C2A and C2B. Okay, now again please tell me which one will be the larger fragment and which one will be the smaller fragment. So the larger fragment will be C2A and the smaller fragment will be C2P. Okay, so larger fragment will going to take part in the next step of that reaction and the smaller fragment will going to dissociate. Okay, so here we have two important parts, C4b and C2a, that will form complex together, C4b C2a, and this complex is known as C3 convertase. Okay, this C3 convertase will bind with the cell membrane of the foreign cell. Okay, so that is the entire concept of classical complement pathway. Although the pathway is not ends here, okay, but as I have told you in the ending of that second video, that the next steps are similar with the lectin pathway and alternative pathway. So I will going to mention the next steps after the completion of the lectin pathway and alternative pathway. So this was the end of this video. So I hope this video will be helpful for you. Thank you.